Hello, everyone, and welcome to today's webinar, Making Therapeutic Antibodies Simple, Fast, and Safe. I am Marie Stone of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by GenScript. To learn more, visit genscript.com. We encourage you to participate today by submitting any questions you may have during the presentation. To do so, simply type them into the Ask a Question box and click Send. We'll answer as many questions as we have time for at the end of the presentation. You may also submit any technical issues here as well if you have trouble seeing or hearing the presentation. I'd like to now welcome our speaker, Dr. Ladan Ding, Head of Antibody Department, GenScript. Dr. Ding, you may now begin your presentation.
Hello everyone, my name is Lidan Ding. I'm come from GenScript. It is my great honor to be here and sharing our experience on the therapeutic antibody discovery projects. Firstly, I will briefly introduce GenScript. Here is the history and milestone of GenScript. From the roadmap, GenScript is keeping actively to develop more technology platform. GenScript was founded in New Jersey, US, 2002, and then we established Europe and Asia Pacific division to facilitate global service in 2019. And last year, we successfully established a new R&D and production facility in Singapore, and a new cell engineering and virus production center in Shanghai, and a new high-throughput facility in US. And on the other hand, specifically for the antibody service platform, we established the customized antibody service in 2004, and until now, we have gained more than 18 years experience on antibody discovery. Back to today's topic, for therapeutic antibody discovery, GenScript can provide one-stop solution from antigen design, gene and peptide synthesis, protein production to antibody discovery. We can also provide antibody engineering such as humanization, affinity maturation, GMP-grade CDMO service, and in vivo and in vitro pharmacology and characterization of antibodies. Today, we will focus on the therapeutic antibody development by rabbit. Then, the first question is, why do we choose rabbit as the host animal for the antibody development? A desired antibody should have high affinity and high specificity, which rabbit is able to satisfy by its nature. In this figure, more than 1,000 MAB were evaluated with affinity by SPR analysis, and the result shows that the KD value of rabbit MAB is mostly around 10 to minus 10 and 10 to minus 11, even 10 to minus 12. However, the affinity of mouse MAB is usually lower, around 10 to minus 8 and 10 to minus 9. That is to say, the rabbit monoclonal antibody performs better affinity when compared to mouse monoclonal antibody. The secret of rabbit have a higher affinity is mainly due to its unique and robust immune system, except antigen-independent B-cell maturation in bone marrow and antigen-dependent B-cell maturation in spleen or lymphocyte. Rabbit has a special secondary antigen-independent B-cell maturation in gut, which only occurs in rabbit and bird. Additionally, rabbit has a longer evolutionary distance with human. Antigens share highly homology between human and mouse could also give a better immune response in rabbit, but not in mice. Moreover, because of the larger body size, except spleen, we can also obtain immunized B cells from lymphocyte, peripheral blood, and bone marrow. Since most of the mice we use in lab were obtained by inbreeding, rabbits with limited inbreeding would also give wide antigenic recognitions after antigen immunization. Here is a table comparing the rabbit and mouse monoclonal antibody. Except the higher affinity due to the special secondary B-cell maturation, there is only one IgG subtype of rabbit. The signal peptide is very conserved. The successful rate of antibody sequencing is relatively high, and the antibody sequence engineering is more easier. Also, one additional diphosphate band exists in rabbit IgG, giving a more stable characterization of rabbit monoclonal antibody, but it is also caused a challenge for the following humanization of the rabbit MAB. Additionally, the epitope recognition ability of rabbit monoclonal antibody is about 5 times higher than that of mice. There are more than 5 isotypes of CD1 molecule. The antigen presenting molecules for lipid, saccharide, and other small molecules. Therefore, antigens, especially small molecules like saccharide, glycolipid, and so on, can also have better immunization performance in rabbits. Rabbit is also preferred to be used for developing mouse, human, and cynomolgus cross-reaction antibodies. In the year 2019, FDA approved the first humanized CFA antibody drug derived from rabbits. This antibody drug was for the treatment of wet AMD, and this drug was owned by Novartis. Due to the high affinity of rabbit MAB, the delivery dosage and treatment frequency of antibody drug were both improved. In the year 2020, the first humanized full-length rabbit MAB drug was approved by FDA. It is a CGRP blocker for migration prevention. This is the first full-length rabbit MAB was used for antibody drug treatment. This message gave us a very exciting and promising signal that full-length rabbit MAB can also be used for therapeutic. Up to now, at least 3 humanized rabbit MAB drugs are in clinical phase 2, and several are in clinical phase 1, such as the antibody against CD40. However, the humanization of rabbit MAB is still in bottleneck. But in the late future, we believe that we definitely could get through and enjoy the benefits of rabbit MAB for antibody drug discovery, because the advantages such as lower dosage, decrease the drug frequency, and weaker off target. The development route of each antibody drug is very long. Then we have to struggle some ways, finding the most fast and latest technology to support and accelerate the antibody development. The main pain point for the antibody drug discovery pipeline is the timeline. With continuous innovation and efforts, now we have successfully established the rapid antibody discovery platform and the fast high-throughput protein and antibody expression plus purification platforms. Then let's have a general comparison between the classic platform and our rapid platform for monoclonal antibody development. Traditional step is in this figure, and the process includes immunization, cell fusion, and screening, and subcloning and antibody expression. Then for each step, what we have done to accelerate the process. Firstly, we developed express immunization method, and we can finish this immunization within 1 month. And secondly, followed by B cell enrichment. The primary B cells will be cultured for about 10 days in vitro, and the function of the secreted antibody will be screened directly by primary B cell supernatant, and this culture process will only take 2 weeks. Thirdly, we will deliver antibody sequence instead of the hybridoma cells, so we can use transient expression or stable cell line generation in a mammalian cell line with our fast protein expression platform to obtain the purified antibody as soon as possible. In total, about 5 to 7 months will be needed for antibody development with fusion method, but with our advanced technology, we only need 2.5 months. And for immunization, we have multiple strategies to solve different types of antigen. We can use recombinant proteins and synthetic peptides as immunogen. For antigens, we cannot get purified protein, such as GPCR targets. We can use DNA or mRNA for rabies and mass immunization. And also, we can use the overexpressed stable cell line and the VLP as the immunization material to obtain a good serum titer. We now have established two rabbit monoclonal antibody development platforms. First one is the B-cell cloning method, and the other one is the single B-cell platform based on Beacon. To save time, all these two methods are fusion-free methods. For B-cell cloning method, the first step is animal immunization. If the immunogen is protein or peptide with high antigenic, we suggest to use the Phoenix immunization method, which only take 4 weeks for the immunization. And once the immunization was done, if the serum type can meet the criteria for the antibody development, we will collect some peripheral blood for the rabbits. PBMC will be isolated from the blood and used for the antigen B cell enrichment. We will directly culture the enriched B cells with the 96 well plates, and we can culture the primary B cells in vitro for about 10 days. We will evaluate if the antibody secreted with the B cell clone is positive to the antigen or not. With high throughput ELISA or FACE. Even more, we will extend the culture of the primary B cells in another 2 days, and more than 1 milliliter supernatant could be collected for further antibody validation, such as the FACS, HC, and ICC. And after assay, and after the different assays of antibody validation, we can further move the positive clones into the antibody sequencing step. And after antibody sequencing, we will obtain the viral region of the heavy chain and light chain of the parental clone. And if more than one variable chain and light chain was obtained from the parental clone, we will do recombination expression of all the variable regions of the VL and VH, and we will collect the supernatant of the recombination antibody, and for further validation by the counter screening, by the positive and negative screening of the antibody. That is to say, the antibody sequencing we delivered was the validated sequencing, and we can finish all the process in industry leading time, the 11 weeks. For Beacon, the single B cell platform, the animal immunization and the enrichment of the B cell is just as similar as to that in B cell cloning. The different step is the antibody screening. The enriched B cell will be directly loaded on the chip after cell stimulation. This is an enlarged view of a part of the chip. There are over 10,000 nanowells of one chip. The machine can recognize the single B cell and then pin into every nanowell of the chip. The function assays will be conducted on chip, such as bead-based and overexpressed cell line-based antigen-positive assays. After the validation of the single B cell, the desired cells with specific ID will be exported into iron-lysate buffer, and the single B cell will move to NGS sequencing to obtain the viral region of the light chain and the heavy chain. Although experiment process can be observed by Beacon, the viral region of light chain and heavy chain can be guaranteed from the same single B cell. That is to say, the viral region of light chain and the heavy chain was natively paired. And then I will take 4-1BB as a case study to review more details of these two processes. 4-1BB, also called CD133, it is the tumor lysis factor and widely expressed on DC-activated monocytes and NK cells. The major biological ligand, 4-1BBL, is expressed on the activated antigen-presenting cells, the APC cells, including DC cells and macrophages, as well as B cells. Ligation of 4-1BB can activate NF-kappa-B and MAP signal pathways, finally contribution to the secretion of IL-2 and F-N-gamma, and upregulation of BCL2 family members, BCL-XL. Therefore, 4-1BB agonists offer an ideal candidate for monoclonal antibody therapeutic target. There are 4 modes of action types of anti-4-1BB therapy. First, agonistic anti-4-1BB MAP therapy induce T-cell proliferation and cytokine segregation. And second, on NK cells, stimulation of 4-1BB can enhance ADCC. And third, agonistic anti-4-1BB MAB can also stimulate DC and macrophage to induce anti-tumor immune response. And also, 4-1BB can expressed in the tumor-associated vascular endothelium cells. And the fourth one is 4-1BB can be function as one part of the biospecific antibodies, such as biospecific antibodies targeting both 4-1BB and tumor antigens such as HER2 can lock immune effectors and target cells in closely. Up to now, more than 100 pipelines are focusing on 4-1BB target, including ABL, Roche, and other pharmaceutical antibodies. Multiple 4-1BB targeted biospecific antibodies are in preclinical or early clinical development and have demonstrated promising early data. 4-1BB is a transmembrane protein, and its extracellular domain has 163 amino acids. For antibody development, due to high homology of cyno and human, it is easier to obtain double positive antibodies. Because only 57% homology is between human and mouse, so it will be a little bit challenging to obtain antibodies with cross-reaction of these 3 species. For antibody discovery, for this project, we use purified 4-1BB ECD as the immunogen for rabbit immunization, and we want to try to obtain higher affinity antibodies with 3 species cross-reaction: the human, homologous, and the mouse. Express the immunization method was used for this project. We finished the immunization within 1 month, and after that, we collected 30 milliliter peripheral blood from 2 rabbits and pulled the PBMC together for B-cell enrichment to increase the B-cell diversity. With the B-cell cloning method, we obtained more than 100 ELISA-positive clones with the protein. And after the confirmation of the facts with the overexpressed cell line, we obtained 43 double positive clones that is positive to overexpressed human and cyno 4-1BB. And at last, we obtained 9 parental clones for the shows 3 species cross reactions. These 9 clones was positive to human, cyno, and mouse 4-1BB overexpressed cell line. And to further confirm of the antibody diversity, we moved the 107 positive clones for NGS sequencing. And after the NGS sequencing, we do the sequence analysis for its for its diversity and pick unique clones for the antibody expression. And 43 clones for antibody was used for the antibody transient expression. And we collected the supernatant and do the further validation of the recombinant antibodies. And in the end, 33 clones was still positive with both the ELISA and FACS validation. And for Beacon platform, after one round of antigen assay positive screening, we obtained more than 1,000 positive clones. And also when we use 4-1BB overexpressed human 4-1BB overexpressed cell line as the screening material, we identify over 200 positive clones that can have binding affinity with binding activity with the overexpressed 4-1BB cell line. And we pick 50 clones for the NGS sequencing. And after the antibody sequencing, we obtained 31 clones that can successfully paired with the VH and VL. And this is the narrowed down process to obtain our target antibodies via B-cell cloning method. After B-cell culture, we firstly screened the supernatant with 4-1BB protein by ELISA, and then FACS was used to further confirmation of the supernatant with the overexpressed cell line. All these assays were performed with the primary B-cell supernatant. That is the most advantage of our platform. We can know which clones was the most shows the most promising data, or which clone is our targeting clone, and then we can move this promising clone for the antibody sequencing to save time and cost. And this is some FACS sample. When we get this result from the B-cell supernatant, we can see that just with the B-cell supernatant, we can identify if this protein clone was positive to different screening material. And to check, this is the detailed antibody sequencing results. To check the antibody diversity and choose the unique sequence for further antibody expression, we applied NGS sequencing for all these 107 ELISA positive clones. We analyzed the similarity of the antibodies and the sequence between each clone. And from the result, we can see that only 5 clones show identical variable sequence. The genetic tree analysis can help us pick the unique clones for further expression. And moreover, we can do more analysis for the antibody pools. Take these 2 figures as an example. We can know how many germlines were obtained in this project, and the germline analysis can do a favor for the further humanization. And from these below 2 fingers, we can get information about the CDR length of each antibody. Then we can pick candidates which can meet the different criteria for the further antibody development. And we then pick up 43 clones for antibody gene synthesis, and we want to see the recombination successful rate of the NGS sequencing, and we want to get the purified antibody for the further functional validation. The recombination antibodies showed consistent function with that of primary B cells. We tested the we recombinant of all these 43 clones, and after the validation with the recombination supernatant, we got 33 positive clones still with ELISA and both FACS validation. And the successful rate of the recombination is around 17%. And then we take one clone as the sample to show that the recombination antibody still shows the affinity binding activity with the 3 different overexpression cell lines: the human form BB cell line, cyno form BB cell line, and both the mouse form BB cell line. This slide shows the detailed assays we conducted on chip at the Beacon platform. This above figure is for the beads-based antigen positive screening. The antigen will be labeled with spelling and was incubated with the assay conjugated beads, and then the antigen will be coated with the beads. And to identify if the antibodies secreted by the simple B cells was positive to this antigen, we will load this assay beads onto the chip in the channel, and we will take photos at different time points, and then the beacon will form a video, and from this video we can know which B cells with specific ID number is our targeted B cells. And after the functional screening, we finally got more than 1,000 antigen beads positive hits. And the below figure is the cell-based assay. When we use the overexpressed cell line with our target antigen instead of the assay beads, we can know the secreted antibody by the single B cells was the cell line binding positive or not. And after this cell line functional screening, we finally got more than 100 cell line positive hits. And then we selected 50 double positive clones for NGS sequencing to obtain the antibody sequencing and analyze the diversity of the antibodies derived from Beacon. We can see that out of 31 clones out of 31 clones, 4 clones show the identical variable region of heavy chain with other clones, and 2 clones show the identical variable region of light chain with other clones. And the successful rate of the NGS sequencing is a little bit lower compared with the B cell cloning, because the sequencing is for the single B cell. The mRNA will be relatively lower. The amount of the mRNA will be relatively lower compared with the B cell cloning. And from this case, we can know that the successful rate of pairing sequencing is around 30%. Because in the B cell cloning method, the cells can be grouped into one B cell clone, and this clone contains more than 1,000 B cells. So the successful rate of B cell cloning is nearly 1,000%, and this is different. The antibody sequencing rate, successful rate, is a little bit different between these two methods. And humanization, after the antibody sequencing, after we obtained the antibody sequencing, another important step is the humanization. Between we can do some functional screening as for the preclinical study, and especially for rabbit, the humanization is very special and critical. And the feature of therefore our company have developed a very special platform for the humanization. It is the AI-based technology, and the general workflow is that we will use the selected template human antibody sequence against the human database, and then use bioinformatics strategy to humanize the antibody sequencing after the humanization generated by AI. And after the humanization, a large antibody sequencing pool will be obtained. So for the candidate humanized antibody sequencing, how can we do selection for the best humanized antibody sequencing? To answer this question, we will also use the AI-based humanization ranking, and then we will select top 10 humanized antibody for the recombination. And after the recombination of the humanized antibody, we will collect the supernatant and do validation of the antibody with ELISA binding method, and we can know if the humanized rabbit monoclonal antibody was still positive to the antigen or not. We can use ELISA FACS or other method for the confirmation. And after the validation of the binding affinity of the binding activity between the antigen and the humanized rabbit MAB, we can further measure the KD value of the humanized antibody with the food bio or BIAC method. And the advantages of the humanization in our platform is that because we use AI assisted method for the whole process, so the turnaround time and the searching scopes will be maximally saved. As we know that the rabbit monoclonal antibodies have many specific characterizations different from mouse and human. The most important point is the C1080 at the viral region of the light chain. On the country region, the C10-171 can form a diphosphate band with this C1080. Therefore, during the humanization, the country region will be replaced with a human sequence, and there is no C10-171, and the diphosphate band of the C1080 will be free. Then this free C1080 needs to be mutated into other amino acids. Except this point, the EF loop, the D loop, and the C10 pairs also should be considered into the humanization. We have developed the rabbit monoclonal antibody humanization, and these are SPR analysis results of our monoclonal antibody before and after humanization. Before the humanization, the KD value is about 10 to minus 10, and after humanization, the KD value is around 5 times to 10 to minus 9. The affinity is reduced about 50-fold. And during the antibody drug discovery, another important step is to develop anti-ID antibodies, which is commonly used for pharmacokinetic analysis in preclinical practice. PK study is the most important step during the, it's most important for the drug metabolism throughout the body, such as like to determine the rate of drug absorption, distribution, and excretion in various cohorts. Researchers need to be able to track antibody drugs which are bound or unbounded to their designated target at various time points post-delivery. To achieve this aim, antigen blocking and antigen non-blocking anti-antibody should be developed for the PK analysis. And we have rich experience on anti-ID project, and this figure is a case study of developing blocking antibodies. We firstly obtained more than 100 clones positive to the immunogen, and this positive, and then we further confirmed this positive clones with the screening material, because the anti-ID antibody will be finally used on the detection of the antibody drug in the human serum. So the class do not want the antibodies shows the cross reaction with the human serum. So we use the total human IgG as a screening material. And after the confirmation, we can see that more than 100 positive clones was still exist, and these clones was positive to the immunogen, to the full length and fiber of the antibody drug, and do not cannot cross reaction with the total human IgG. And for this project, because the class want to generate a blocking antibody, so after the binding of the antibody with the antibody drug, we do a competitive ELISA to select blockers for the class, and finally we obtained 11 clones that shows higher blocking activity between the antibody drug and ligand. After the lead generation, the following question is: how can we evaluate the drug safety? We can provide PK and ADK development to measure antibody drug concentration in different metrics, such as serum and neura, with high sensitivity and a larger linear range. We can also provide HCP kit development for the measurement of the protein of the host cells that be used for the antibody expression. For all the kit projects, we could offer from raw development to GMP grade production. In summary, we have established the highly customized, scalable total antibody drug discovery solutions at different stages, including but not limited to gene and peptide synthesis, virus packaging, antibody development, and lead generation, antibody production, and kit development. Now, GenScript is a world-leading biology CRO company with more than 5,000 employees, serving more than 30,000 customers from more than 100 countries. We have more than 18 years experience of antibody development and have delivered more than 150,000 antibody projects. We have the most latest technologies to maximize your success at achieving high-quality antibodies. And also, for the ownership and the IP are totally owned by customer for the discovered antibody. My presentation will stop here, and it's our pleasure to share the antibody discovery story of ours. We're more than happy to be a partner of you and speed up your research.
Thank you, Dr. Ding, for your informative presentation. We will now start the live Q&A portion of the webinar. If you have a question you'd like to ask, please do so now. Just click on the Ask a Question box located on the far left of your screen. We'll answer as many of your questions as we have time for. Let's get started. Our first question is, what do you mean about the ALAC and OLA animal facilities?
Thank you for this question, and I will answer in the following words. And OLA is short for Office of Laboratory Animal Welfare and certificated by DHHS and NIH in US. And the OLA provides guidance and interpretation of the public health service on human care and use of laboratory animals. And the OLA also supports educational programs and monitors compliance with the policy to ensure the human care and the use of the animals in PHS supported research, testing, and training. And the ALAC is the full name of Association for Assessment and Accreditation of Laboratory Animal Care. It is a non-governmental and non-profit international certification organization, and it is also the certification for animal breeding and animal-related experiments. The ALAC require scientific treatment of animals in the fields of biological science and medicine in order to ensure and promote the quality of animal experiments. The US FDA and the Europe commitment strongly recommend animal experiments was conducted in ALAC certificated laboratories. And in summary, in the United States, if you come from the United States and want to apply some project funded by the NIH, FDA or CDC, maybe the OLO certification is required for the animal facility. And the ALIC is a global certification organization for animal welfare requirements. And this is my answer for this question. And welcome to more questions.
All right, thank you. Next question, how to compare two different rabbit MAB and development technologies, single B cell and B cell cloning?
As I have present during the webinar, for the B cell enrichment and the animal immunization is just as similar as in both single B and B cell cloning method, and the main difference is about the antibody screening step. For the Beacon platform, because we call it the single B cell, so the most advantage is that all the assays and experiment can be conducted at the single B cell level. So the Beacon is a very most comprehensive machine for the function to detecting the function of the secret antibody. And the detail is that the cells can be penetrated into every nanowell, and the single B cell can be observed before and after the cells would penetrate into the nanowell. And also we can do the function screening on the chip after the pinning was done. We can both use the antigen-positive bead-based antigen-positive assay, and also we can use cell line to identify if the antibody was cell line positive or cell line binding antibody. And we can do several assays in one day. For one round of screening, maybe 15 to 30 minutes will cost, and that is to say, we can do at least 3 to 5 rounds of assay screening on the chip in one day, and so it is a very comprehensive machine. And because the before and after the B cell screening, all the experiment can be observed, so we can guarantee the viral region of the light chain and heavy chain was come from the same B cell. Means the viral region of light chain and heavy chain was natively paired. But for B cell cloning, because we cultured the B cells in vitro after the B cell enrichment, and we dilute the B cells with the serial dilution method. Due to this method limitation, we want most of the wells was a single B cell clone, but after the growth, after the culture of the B cells, maybe more than one clone will obtain. So maybe more than one variable region of heavy chain or light chain will be obtained. But we will do randomly recombination of all these variable VH and VL, and do validation from the recombinant supernatant. So even though we cannot observe that the VH and VL was from the single B cells, but because we want to maximally guarantee the cells was a single B cell clone, so we can relatively to say that the viral region of light chain and heavy chain was natively paired. But if you sensitive to this point, maybe Beacon will recommend it. And this is the answer for this question.
All right, thank you. Next question, what other antibody characterization can GenScript perform?
For the antibody screening, ELISA and FACS was the most used method for the antibody screening, except for this two high throughput screening method. We can also do other functional validation screening, such as the cell-based ELISA and cell-based HC. These two methods can help us to find the suitable clones that can application in the HC, and also we can provide ICC and WB, IP pull down, and so on, and some other assays to select positive clones that's suitable for your down application. And with the purified protein, if we express the antibody, and after the purification of the antibody, we can use the antibody. We can use the HPLC mass for the quantity control. And if you want to measure the KD value, the affinity of the antibodies, we can perform the affinity measurement with football or the BRCO, the BLI or SPR methods.
All right.
That's all.
All right. Next question. What are the key advantages of choosing GenScript's antibody services?
Oh, thank you for this question. And I think that there are truly some advantages that help you to choose GenScript as the first choice. Maybe the first point is we can provide one-stop solution from gene and peptide synthesis, protein expression, antibody discovery to lead validation, even the GMP level kit production. Because we are a very comprehensive one-stop solution CRO company. And the second is that because of the fee-for-service model for the customized service, all the IP, all the ownership of the project deliveries, such as the antibody sequencing, was totally owned by class. And also from the history and milestone, we can see that we have rich experience on the reagent product development and also the antibody development. We have more than 18 years experience and have finished more than 150,000 projects in total. And also, as I have answered in the first question, we have the ALIC and OLO double certificated animals facility. And for the technology, we have developed some self-developed and most latest platforms to support the basic research and to maximize your successful rate. For example, we have the coding optimization tools and the anti-aging design tools to help to increase the protein or antibody expression level, and also to design the best antigen that can be used for immunization. And also, if the homology is very high, for example, even 100% between human and mice, we have the ImmuPlus technology to break the tolerance for the immunization. And also, as shown in the webinar, the turnaround time of our service for the antibody development or the protein production, with the turnaround time is short, is industry leading time, I think. And also, except the single B cell, the Beacon platform, and the B cell cloning, we also have other antibody development strategies, such as the traditional hybridoma fusion method and the phage display method. And I think this is all I could summary for this question. If if you want to learn more, I think you can contact us literally or find some information from our website.
All right, great. Thank you. Next question. Is a 4-week immunization in rabbits adequate in all cases?
So can you repeat your question?
Yes. Is a 4-week immunization in rabbit adequate in all cases?
Oh, thank you for the question. The 4 weeks immunization is our self-development method. For rabbits, the process the whole process will take 4 weeks, and for and the whole process was previously settled down. We will just if the first injection was given to the animal, all the timeline will be settled following the first injection. And also we have, I want to say here, we have the expressed immunization method for mouse, and it will only take 2 weeks.
All right, great. Next question. In addition to affinity, will other factors, such as heterogeneity, be considered for choosing the top clones?
Yes, I know that the high successful rate of the positive clone is good, but also it is a little bit challenging for clients when they facing a plenty positive clone or antibody pool. They want to know which clone was the best for their demo application. So my answer is except the affinity. Maybe you should consider about consider about your down application. For example, if you want to generate an HC antibody for the in vitro detection, except the affinity, maybe the low background should be considered. So we can do some counter screening during the antibody development. And for the antibody drug development, because most of the antibodies will be used in the clinical clinical experiments. Maybe you want the very low cross reaction of the antibody with the metrics such as the human or animal serum. So except the affinity, maybe we can add the serum as the constraining material, and I mean the to narrow down the antibody tool. And if you want to detect some small molecule or some proteins during the metabolism, so the sensitivity of the pairing antibody should be considered. So you should we will select. We will do some, for example, epitope binding to help you select the antibodies that can recognize more than one epitope. So it can increase the successful rate to obtain the paired antibody for the sandwich ELISA for the in vitro diagnostic detection. So I think maybe to narrow down the antibody pool, except the affinity, you should consider more about the application.
All right, thank you. Next question. Generally, how many top clones are recommended, since for single B cloning, over 100 clones are possible?
From the case study I have shown, the more than 1,000 positive clones will be obtained after the one round of screening on one chip. But the export of the cells, so it will every every single B cell will cost about 3 minutes. So the limitation of the Beacon platform is not the portable haste, is about the export time. So we will recommend you to export the top 100 or even 200 clones to the unlicensed buffer. But how can we select this top 100 from these thousands of positive clones? Maybe we can we can design more assays to narrow down the positive list. For example, except the antigen positive screening, we can narrow down the positive haste with the cell line overexpressed cell line screening or some blocking assay screening, and in the end maybe we can recommend you to export about 100 to or less than 500 clones to the analysis buffer.
All right, thank you. Next question. What's our success rate so far for anti-ID projects, and how many unique clones do you expect?
That's the most frequent question we will be asked from class. Or in generally, most of class maybe think that it is more easier to obtain non-blocker or non-blocking antibody for the detection of the total antibody drug. But according to our experiments, we have finished, this year we have finished more than 50 projects of the anti-ID projects, and the successful rate of the blocking antibody is very high. All of the projects we successfully delivered the blocking antibody. That it means 100% success rate is for the blocking antibody development. But for the non-blocker, because the anti-ID antibody was mostly used for the pharmacokinetic analysis, the PK analysis, so the matrix will be complicated, such as the animal serum or human serum. So total human IgG or total human IgG 1 or 4 will be needed for the count screening. We do not want the anti-ID antibody have any cross reaction with the when we detect the antibody drug in the human serum or urine matrix. So after the count screening, most of our anti-ID antibodies that can recognize the country region or the viral region of the FR will be excluded. So the remaining antibodies will be partially or 100% blocker antibody for this project. So for such projects, the non-blocking antibody is a little bit difficult. The success rate is a little bit lower. Up to now, we have there are about 3 projects would fail to obtain the anti-ID, the non-blocking anti-ID antibody. So the success rate of blocking antibody is 100%, and for the non-blocking antibody, it will be a little bit lower and difficult.
All right, thank you. And it looks like we have time for one more question. Will cryopreservation of PBMC affect the efficiency of getting positive cloning?
Thank you for this question. From the workflow, we can see that we use the peripheral blood collected from the rabbit because the body size is very large. For the B-cell cloning, but if the immunization was not done in our company, we also can accept the stored B cells, PBMC cells, or even splenocytes us. But if the initial material is not fresh PBMC, maybe the successful rates will be a little bit lower. According to our comparison data, we get the conclusion that the positive rate will be 50%, will be around 50%. That is to say, about half of the positive clone will be lost due to the cells was stored in the liquid nitrogen and then salt into the room temperature, the cell culture temperature. So the success rate will be 50% compared with the fresh PBMC.
All right. Thank you, Dr. Ding. Do you have any final comments for our audience?
Yes. Because the therapeutic antibodies have become the predominant class of new drugs development in recent years, I think the most of the class want to concern the most is the timeline. The timeline is always the pinpoint during this process. And with our mission of GenScript, we want to make people and nature healthier through biotechnology. We have integrated a series of precise and consistent scalable solutions to facilitate each principal milestones in antibody drug discovery. We will provide a very high speed and quality antibody discovery process towards the IND. And today we take 4MBB as a case study to show our ability to show what can we do to support your work. So here I want to thank you again for everyone to attend this webinar today, and I hope to work with you soon to support your project.
Well, thank you again, Dr. Ding, for your time today and your important research. We would also like to thank LabRoots and our sponsor, GenScript, for underwriting today's educational webcast. Before we go, I'd like to thank the audience for joining us today and for their interesting questions. Questions we did not have time for today, and those submitted during the on-demand period, will be addressed by the speaker via the contact information you provided at the time of registration. This webcast can be viewed on demand. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, take care, everyone, and goodbye.